Hi, I'm Sean Stevenson, a chemist on the Decontamination Sciences branch at the DevCom Chemical Biological Center. Today I'm going to share with you the work I have been involved with exploring the use of zirconium hydroxide for the decontamination of chemical warfare agents from skin. To provide a little background on the program, our team was funded by the Defense Threat Reduction Agency in 2015 to look at the ability of common off-the-shelf items to decontaminate chemical warfare agents from skin. Think things you can buy at Target: shampoo, body wash, etc. Around the same time, zirconium hydroxide was coming onto the scene as an absorptive powder that was reactive toward chemical warfare agents, so we threw this technology into our evaluations as well. Our first studies looked at the efficacy of these items on Strat-M, a synthetic model for transdermal diffusion. Turns out zirconium hydroxide was one of the top performers. Moving into 2017, we found that zirconium hydroxide showed good efficacy on contaminated pigskin as well. Concurrently in 2018, Dr. Chilcott and his team at the University of Hertfordshire published the PRISM studies, which looked at the efficacy of different decontamination treatments on human volunteers. One of the notable findings was that the use of a dry decon step prior to anything wet improved the overall efficacy of the treatment. Being that we were researching zirconium hydroxide as a dry skin decontaminant, a collaboration between our groups seemed useful. So fast forward to 2019, when a formal collaboration was established between DevCom CBC and UH to mature the skin decontamination technology. So what is zirconium hydroxide and how does it work? It's a common powder found in a variety of things like antiperspirants and inks. Its utility in neutralizing chemical warfare agents comes from its basic terminal hydroxyl groups and the acidic bridging groups. It neutralizes GD and VX quickly, but HD takes a little longer. Now that we have this powder that shows promising decontamination efficacy on skin, what's next? One of the main tasks was to explore the efficacy further. We know the powder provides agent removal from skin, but how does this compare to other techniques? Is it effective at different agent-skin interaction times or decontaminant residence times? Does the powder provide efficacy in an in vivo model? For today's brief, I'm going to focus on the top bullet. My team, along with the University of Hertfordshire, used novel diffusion cells with agent and simulant to compare the performance of the zirconium hydroxide to a shower in controlled condition. For these tests, we use a diffusion cell setup. If you're not familiar, a simple diffusion cell consists of a membrane, like skin, sandwiched between a donor chamber and a receptor chamber. The agent gets dosed onto the membrane in the donor chamber, then you measure the liquid in the receptor chamber to determine how much and how fast the agent diffuses through the membrane. University of Hertfordshire developed a specialized version of the diffusion cell, which allows for all kinds of decon treatments to be investigated. The cell allows for a greater surface area of the membrane to be exposed, for a shower to be applied to the membrane, and for the sampling of other areas than the receptor chamber, such as vapor off-gassing from the skin during the experiment. So, with setups of these modified cells at CBC and UH, we completed testing looking at agent and simulant decontamination efficacy of showering or zirconium hydroxide application in an immediate decontamination scenario. A control condition with no decontaminant was also included. UH did the simulant work and we did the agent work. Our methods and techniques were identical except for our analytical methods. UH uses radio-labeled simulants measured via liquid scintillation counting. We measure neat agent via GCMS and LCMS after extraction from various test samples. One thing to note with these differences is that the liquid scintillation counting measures the radioactivity of a sample but does not differentiate between the parent molecule or a reacted byproduct. Our methods look for a specific analyte. This comes into play a little later when I show you the data. So let's look at the results. First up is HD and its simulant methyl salicylate. What you see here is the percentage of the applied dose for each treatment-agent combination. Each color represents the location where the agent was found at the end of the treatment. First thing to note, and it is more obvious on the zoomed-in right-hand figure, is that more HD was found in the pigskin, meaning using methyl salicylate as a simulant will underestimate the dermal penetration of HD. For showering and zirconium hydroxide treatments, both were very effective at limiting the HD that was absorbed into the skin. Another thing I want to point out is that we were unable to fully quantify the massive HD that was in the shower effluent, hence the low % recovery. HD is not super soluble in water, and we tried a variety of techniques to accurately measure the HD concentration, such as liquid-liquid extraction, adding salt, etc. So while it looks like we did not recover all of the initially applied agent, we're pretty confident that the difference is hiding in the shower effluent. Moving on to the results of GD and its simulant diethylmalonate, you have the same figures as the previous slide. Across all treatments, more GD was found in the skin than diethylmalonate. This was expected due to the differences in vapor pressure and log P for each chemical. Some notable data to showcase here is that the substantial GD off-gassing in the control samples, but not in the zirconium hydroxide samples. This indicates that the powder greatly reduces any GD respiratory hazard. This data also illustrates the struggle we had working with zirconium hydroxide. Not only is it absorptive, but also reactive. So when we go to extract the powder for residual GD, we're fighting with it being tightly bound and hoping to pull off reacted byproducts as well. We know the GD reaction with zirconium hydroxide forms PMPA, so we look for that in our extracts as well, but even then we were not confident we were recovering everything from the zirconium hydroxide. We do have confidence in the rest of our measurements though, so it's assumed that the missing agent is tied up in some shape or form in the zirconium hydroxide. Lastly, here are the results for VX and its simulant 4,8. Like HD and GD, more VX was absorbed into the skin than its simulant. The VX case was also the same as the GD case. Zirconium hydroxide worked well at keeping VX from getting into the skin, but was difficult to accurately measure in the powder due to the strong absorption and reactivity. So, to summarize, methylsalicylate, diethylmalonate, and 4,8 can be used as simulants for HD and GD and VX, but they will underestimate the amount of chemical warfare agent that absorbs into the skin. Also, the performance of zirconium hydroxide was just as good as showering, but provides a key logistical advantage. No water is needed. This is especially useful for a warfighter who is downrange, who needs to quickly decontaminate and continue the mission. The diffusion cell work is just one piece of the entire program, and other tasks have been completed or are ongoing as well. Naveen Amer from the University of Hertfordshire has a poster presenting temporal studies completed with zirconium hydroxide. Devanya Mahalingam from University of Hertfordshire has a poster presenting our efforts to validate a robot for use in exploring how to best apply Decon technology. And Andrea Peenhall from the University of Hertfordshire has a poster presenting efforts to understand the dermal absorption of zirconium hydroxide into skin. With that, I'd like to thank Dr. Bass and Dr. Higgins at the Defense Threat Reduction Agency for funding this work, all my excellent colleagues on the Decontamination Sciences branch for their help with planning and testing, and the team at UH for their fantastic work as well. Thank you so much for your time.